Plant tissues. We have already learned that there is a difference in the growth of animals and plants. In animals, there is no such demarcation of dividing and non-dividing regions. They have uniform growth all over the body. While growth in plants is restricted to specific regions, that is, the tips of the roots and stem, where dividing tissues are present. Let us perform an activity to understand about plant tissues. Activity. Take two small gas jars, label them as A and B, and then fill them with water. Now take two onion bulbs and place one on each jar such that the lower portion of the onion bulb is immersed in water. Observe the growth of the roots in both the bulbs every day. Measure the length of the roots on day 1st, 2nd, and 3rd and note it down in the observation table. On day 4, cut the root tips of the onion bulb in jar 2 by about 1 centimeter. Now, observe the growth of the roots in both the jars and measure their lengths each day for 5 more days. Record the observations in the observation table. From the observations, we can say that the onion bulbs in the first gas jar, that is A, have longer roots. Why does this happen? The roots of jar A are longer since they have dividing tissues at the root tip. Thus, these roots grew continuously and their length increased. But in jar B, there will be no growth of cut roots since the dividing tissues are absent. Now, let us understand in detail about dividing tissues. Meristematic tissues. Meristematic tissue is the only plant tissue that produces new cells by cell division. The meristematic tissue are present at specific areas and thus growth is restricted at those areas. The cells of this tissue have prominent nuclei, dense protoplasm, that is, cytoplasm. The cell walls are thin and arranged in a closely packed structure. Most of the cells do not have vacuoles. The cells of these tissues are active. The main function of the meristematic tissue is growth of the plants. Depending upon the region where they are situated, they are classified into 3 types as apical meristem, intercalary meristem, and lateral meristem. Apical meristem. These tissues are located at the growing tips of the roots and stems. It enables the root or stem to grow in length. Intercalary meristem. These tissues are located at the base of the leaves at the nodal regions of the twigs. This helps in the growth of leaves and twigs. Lateral meristem. These tissues are located at the lateral parts of the roots and stem. Due to lateral meristem, the girth and the width of the stem or the root increases. We have now learnt about the meristematic tissue. Now let us study the next section. We know that plants are multicellular and made up of different types of cells. Let us perform an activity to understand more about this. Activity. Take a plant stem. Let us cut it into very thin slices or sections. Dip these slices in water. Now, add a few drops of safranin in water and place the slices in this solution for some time. Place a neatly cut section on a slide and put a drop of glycerin. Cover with a cover slip and observe it under a microscope. What do you see? In the slice of the stem, we can see different types of cells. These cells have different structures and are different from each other. This shows that there are different types of cells in the different parts of the plants. The different types of cells carry out different functions. Now let us understand in detail about permanent tissues. Permanent tissues. The cells formed by the division of the meristematic tissue take up a specific role and lose the ability to divide. This process of taking up a permanent shape, size, and a function is called differentiation. As a result, they form a permanent tissue. Permanent tissues are of two types: simple permanent tissues and complex permanent tissues. Simple permanent tissue. Simple permanent tissues are made up of one type of cells. Depending upon the functions they perform, the tissue is classified into the main types: parenchyma, collenchyma, sclerenchyma, and surface tissue. Let us study each of them in detail. First, let us understand about parenchyma in detail. Parenchyma. The cells in these tissues are thin-walled, have intercellular spaces, and are living. These cells are present in the different parts of the plant, like the roots, stem, leaves, flowers, fruits, and seeds. The main functions of the parenchyma is to fill empty spaces to give mechanical support to the different parts and storage of food. Parenchyma are classified into two types: chlorenchyma and arenchyma. Chlorenchyma. The leaf of the plants contain parenchyma. The chloroplast present in them contains chlorophyll, hence the tissue is called chlorenchyma. It performs the function of photosynthesis. Arenchyma. In aquatic plants, the intercellular spaces form large air cavities. They give buoyancy to the plant and help them float in water. Such parenchyma is called arenchyma. After studying about parenchyma, now let us study about collenchyma. Collenchyma. The cells of this tissue are living and elongated. Their cell walls are thickened at the corners with cellulose and pectin. This tissue is located mainly at the base of the leaf, stalks, stem, and branches. These tissues give flexibility and support to the plants. Now let us understand more about sclerenchyma. Sclerenchyma. The cells of these tissues are elongated and narrow, fibrous and dead. The cell walls are thickened due to the deposition of lignin. This tissue is located in the stems, the veins of the leaves, in the hard covering of seeds and nuts, and the outer covering of a coconut. Due to the specific structure, the tissue makes the plant hard and stiff. After studying about sclerenchyma, let us now understand about the surface tissue in detail. Surface tissue. Let us perform an activity to understand about surface tissue. Activity. Let us take a freshly plucked leaf of lily, rio, or any fleshy leaf. Stretch and break it by applying pressure. While breaking it, keep it stretched gently so that the transparent peel of the leaf is seen. Remove the peel and put it in a petri dish filled with water. Add a few drops of safranin. Keep the peel in the dilute solution of safranin for a couple of minutes. Gently transfer it on a slide and place a cover slip over it. Observe the slide under a microscope. What do you see? We can see the outer layer of cells. These cells are the cells of the surface tissue. The entire surface of the plant consists of a single layer of cells called the epidermis. The cells of the surface tissue are relatively flat without intercellular spaces. This creates a continuous sheet. Plants like cactus, growing in very dry habitats have thicker epidermis. Under unfavorable conditions, the amount of water is less. Epidermal cells on the aerial part of the plant secrete a waxy, water-resistant layer on their outer surface. Thus, a waxy layer called cuticle is formed on the epidermal cells on the stem and leaves. Due to this layer, loss of water from the surface of leaves by evaporation is prevented and water is retained. Thus, the layer of cuticle, besides protecting against loss of water, protects the plant from mechanical injury and invasion of parasitic fungi. Hence, epidermis is the protective layer of the plant. Metabolic activities like photosynthesis and respiration need the exchange of gases from the atmosphere. For exchange of gases, the outer surface has minute openings called the stomata. The stomata are present on the epidermis of the leaf. Transpiration takes place from the stomata, that is, water is given out in the form of vapor. The stomata are enclosed by two epidermal kidney-shaped cells. They are called guard cells. They control the opening and closing of the stomata through which transpiration takes place. Now, you must have realized why more stomata are found in the lower leaf surface than the upper surface. If there were more stomata on the upper surface of the leaf, there would be more loss of water. Thus, the amount of water content would decrease, causing wilting of the plant. Hence, there should be an appropriate amount of loss of water during transpiration, which will help to maintain the appropriate temperature of the plant. In hot climate, due to transpiration, the temperature of the plant does not increase. The epidermis of desert plants, like cactus, is different from that of other plants. It has a thick, fleshy outer covering. It also has spines. Due to the dry and hot climate of the desert, to prevent loss of water by evaporation, there are spines on the cactus. Also, the cactus stores water in the stem, due to which its epidermis is thick and fleshy. Let us study the next section. If you touch the trunk of huge trees, you will realize that the bark of such trees is dry and rough. Why does this happen? As woody plants grow older, the cells of the epidermis break and die. They are compactly arranged. Therefore, they do not have intercellular spaces. They also have a chemical called suberin on their walls. Due to this chemical, there is no exchange of gases and water from the bark. The bark of the tree made of dead cells gives strength and support and also acts as a protective layer. The cells of the epidermis of the roots have long thread-like fibers, due to which the surface area of the outer surface increases and absorption of water is done more efficiently. After studying about simple permanent tissues, now let us study about complex permanent tissues. Complex permanent tissues. Complex permanent tissues are made up of more than one type of cells. Complex permanent tissues are of two types, xylem and phloem. Let us study these types with the help of this table. Xylem. Most of its cells are dead. The cell walls of the cells are thick. They are of different types: tracheids, xylem vessels, xylem fibers, and xylem parenchyma. Of these tracheids, xylem vessels and xylem fibers are dead cells, while xylem parenchyma are living cells. The cells of these tissues are in the form of continuous tubes. This allows the transport of water and minerals only in the vertical direction from the bottom to the top. phloem. These tissues are made of living cells containing cytoplasm. Phloem is made up of 4 types of cells: sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Of these, sieve tubes, companion cells, and phloem parenchyma are living cells, while phloem fibers are dead cells. These are connected to one another in the form of tubes. The phloem transports sugars and amino acids from the leaves in the upward as well as in the downward direction. After studying about xylem and phloem, now let us perform a small activity. Activity: let us first take a radish and cut its tip. After you have cut the tip, you will see a white elongated section in the center. Now let us take some water in a beaker. Add a few drops of red ink into it. Keep the radish with its tip cut in the red-colored water standing for about 2 hours. After 2 hours, observe the radish. What do you see? We can see that the central elongated portion has become red. From the types of cells that we have studied earlier, we know that the red-colored tissues are xylem. It is due to the tracheids and xylem vessels, water and particles of red color in it flow in the radish in the upward direction and change the color of the white portion of the radish. So far, we have studied about tissues and the types of tissues. Now, let us understand about biotechnology and tissue culture.